Alright, so today, first off, we're going to be going through DNA replication. Now, the whole point of DNA replication is to use already made DNA to create another section of DNA. So hopefully you'll start with this, and you'll end up with two of these. So the first thing we're going to look at is DNA replication with an example of E. coli as the base. So out of the ingredients that we have all here in this DNA cake, we have DNA, which you need obviously because it's a templated process. You have DNA polymerase 1, 2, and 3. You have DNA gyrase, the sliding clamp, the ATP required for that, and some other processes. You have your DNA helicase to break that apart, your DNA primase to create RNA primers that DNA polymerase 1 recognizes and attaches complementary sequences to. You have DNA ligase, which attaches the Okazaki fragments together from the lagging strand. Single strand binding proteins here that collect on the exposed nucleotides after DNA helicase breaks them apart. You have the DNA A protein, which starts the point of origin, and that's everything that goes into this cake. And then here's a little breakdown of it. You have your lagging strand, leading strand, DNA helicase, your gyrase attached, your DNA polymerase 1, 2, and then 3 here at the bottom with the sliding clamp attached. That's your little breakdown for DNA replication. Now we're going to bake this cake. Alright, so we already got some of the little bit of ingredients cooking. So we can go ahead and add the rest of the pot. And hopefully we'll get some more after this. And would you look at that? 2 completely distinct, separate strands of DNA. So the ultimate goal of transcription is to lead from DNA to pre-mRNA to mRNA. And this is kind of a complicated process, but the first step is to go from DNA to pre-mRNA and then hopefully RNA. And then here's a little trick to get to there. So the entire point is to get the mRNAs from the original DNA strand. So we have this, and we don't need this anymore. This is future Jonathan, by the way. I ended up needing that DNA strand for the next shot, so disregard throwing that and disregard it having a broken section. So this is a very quick and easy explanation of transcription using RNA polymerase. So RNA polymerase is an enzyme that catalyzes this reaction to produce a new strand of mRNA from a DNA strand. So this travels in this direction. You have your Mg2 plus active site here at the bottom, and you actually have a double helix interaction between the RNA strand and the DNA strand using this template strand here, and then this creates the complementary strand from that. So this is an example of transcription elongation. So this here is this double colored sequence right here. This double colored unit is the RNA polymerase. It wraps around the DNA strand, which is going this direction. It's traveling along this way. So this red section right here is the mRNA sequence that's being read off from the DNA sequence. So first you have this, and then you have the CD tail here on the attached to the RNA polymerase where phosphorylation happens to attach the 5' end protein, and then the 3' adenylation that happens after this is cut. You have your specific splicing protein, which is denoted by this little yellow piece right here. And then after this gets released, you have 3' end processing proteins that will eventually end the mRNA sequence. It's often stated that the first step in gene expression is transcription. No, I'm cutting this. Certainly not least, we have translation. So before we can get into the meat and potatoes of translation, we have to talk about tRNAs. tRNAs are synthesized by RNA polymerase 3. These and this are the same thing. This is a covalently modified portion or covalently modified version of this. You can see the similarities. They have the anticodon at the bottom, a 3' end up at the top that is going to be modified by this enzyme, which is a tRNA synthetase. So this specific synthetase adds on an amino acid to the 3' end of the tRNA, which will be later on used in the ribosome. And as you're watching this, you're seeing a real-life reenactment of the process of the attachment of the amino acid by the amino acetyl tRNA synthetase. So now we're inside of the ribosome. So the mRNA sequence is in the ribosome at this moment, and then you have these tRNAs with the amino group attached to it going to each specific codon. Now the first one is the start codon, which is AUG, which codes from methionine. So that one will go and attach to that, and then leave eventually. So we're just gonna pretend that that's leaving. Should have brought a napkin, but that's fine. And so it goes away like this. And then another one attaches here, bringing another amino acid, and then it starts building up the amino acid and forms a polypeptidyl RNA. Hi, voiceover Jonathan here. Regular Jonathan forgot to say that that's a polypeptidyl tRNA. Just wanted to make sure that he didn't mess that up. Thank you, post Jonathan.